Okay, good morning, guys. First of all, go Bolts. We won last night the Stanley Cup. That was fun. Um, Secondly, I wanted to say some things before we got into the review today. So, for this exam block, hopefully you guys have been doing well. Hopefully it's going well for you guys. But if it's not going well, you can always feel free to email me, set up a meeting with me, and try to figure out how to change up your study habits to make it better for the future. If you didn't get like all A's on these exams, it's not the end of the world. I didn't get an A on every exam in the program, so don't think that everyone was like perfect and that you can't come to us if you need help. Because it's better if you come to us like early on and we could try to figure it out with you so that you could like do well in the next two blocks. So, yeah, don't feel like you can't come to us for help. We are all not perfect, so you don't need to be like intimidated by that. Also, we really just want to help you guys. So, yeah, but hopefully that's not the case. Hopefully, you guys are doing well. Um, but I just wanted to let you know that we can help you if it's not necessarily going as well as you had hoped. Okay. So that was one thing that I wanted to say. And then another thing that I wanted to say was some of you, if you've done the practice exam, then you know what I am talking about. Ah. If you haven't done the practice exam yet, then you'll see once you do it what I'm talking about. But Dr. Blank wanted me to put in that first paragraph before the practice exam. Um, it's not that it's not proofread because I did proofread it. He went over it with me. I took like 3 days to write that practice exam because I was trying to write it like pretty similar to how he tested us. But what he was getting at is that I'm writing the practice exam, whereas he's writing your actual exam. So, you can't just take my practice exam and be like, oh, I did well, like I'm done studying. He wants to make sure that you guys are still studying, even if you like take my practice exam and do well. So I tried to put in questions that were like pretty similar to the ones on the test. So that is a good study tool to make sure you're understanding. And if you get some questions wrong on the practice exam, then go back to those topics. But it's not meant to be like, oh, I got an A on her practice exam, I'm getting an A on the test, I'm done studying. And he just wanted to make sure you guys understood that. So that's all that paragraph is about. And then, I wanted to go over. Oh, my iPad is probably dead. I wanted to go over with you guys all of the practice questions that you guys have. It's like a lot. So you get 5 from me every TA session for every lecture. You get 12 every TA session on Fridays, the Q&A practice quiz. And then you got a 50-question practice exam. Then he gave you some practice questions. Then he'll give you example questions in the lecture. Then he gives you the fill in the blanks. So once you add all of those together, that's like 220 practice questions. So if you do all of those questions, then you should be like pretty good, because if you do those questions and you not just know the answer to that specific question, but if you like study the topic itself and you understand why it's right, this is wrong because of this, whatever, then you're going to be pretty good, because the test is only 50 questions and there's only so many things he can ask you questions on. So doing all of those practice questions is like really beneficial to making sure you're understanding and you go into the test prepared. So definitely, if you haven't looked on Canvas and seen my document of what to look over before the exam, do that, because I have it like all broken down of things that you should definitely be studying before you go into the exam. And then I also have like important topics to know on that same document. So definitely look at that. Okay. With that being said, now let's get into why you are actually in my session, the material. Okay. So this might be how you're feeling right now, but it's okay. You don't always have to be a strong cowboy. Sometimes being an alive cowboy is just enough. You're halfway through. You can see the light at the end of the tunnel. And hopefully, like this Friday, you guys can relax and have some fun before prepping for the next block. Okay, so here is a suggested reading only, FYI only. And this translesional polymerase, that's going to be a decoy if it's on the exam. So we don't really need to know that. We're not paying attention to it. It's a decoy. Don't let it trick you. Okay, so for the case, okay, like I had said previously, the cases are lower priority. I went back to my notes to see if I had taken any notes on the cases or answered like these case questions or anything, and I never did. And obviously I did well in the course, well enough to become your TA. So I wouldn't say that it's necessary, but I'm also not writing the exam, so I don't want to say that and then you guys get mad at me if there might be a question. But I literally never answered these case questions, and I never studied the cases. He even said it in class. It is lower priority, so I wouldn't waste my time with it if I were you guys. I would make sure I'm studying all the material, making sure I'm doing all of the practice questions, and then if you have some time, look them over. And if you are going to look over the case and case questions, I would start with this one because he references this one a lot in the lecture. But these cases, I think, are more of just, this is a medical science master's. I'm going to incorporate something medically related to try to keep them interested. But since it is related, you know, we're going to talk about it in the lecture. So the questions are coming from the lecture, what he actually spent his time on discussing in class. So take that how you will. Yeah, so now let's just like get into the actual lecture. So, okay, we start off by talking about types of DNA damage. So this is not really our focus for the whole lecture, but we have to understand the DNA damage, the different types, whatever, how it's occurring, to understand the repair mechanisms that are in place to fix this type of damage. So we can have mutations. So we can replace one base for another. And then we can have thymine dimers, which are caused by UV light. They're very common. It's a covalent connection of two thymines next to each other, and it's a target for repair processes. So I'm fairly certain everything we've discussed, besides the phosphodiester bond, glycosidic bond, and hydrogen bonds between the bases, everything besides those three have been covalent connections, covalent bonds, whatever. So I don't necessarily think he will test you on that, but just keep that in mind. And mutations can cause a significant distortion of the helix or like not as significant of a distortion in the helix. But when we have thymine dimers, we're always going to have a distortion in the helix. And it's the distortion that is actually going to be stimulating the DNA repair. That's what this little S is, stimulating. Okay, so we know what our normal Watson-Crick base pairing is, G to C, A to T, and here you can see we have our hydrogen bonds between them. But if we add methyl groups, then we can disrupt our normal Watson-Crick base pairing, which is what we're seeing on this slide. So methylation of the guanine prevents cytosine from base pairing and permits thymine from base pairing. So instead of having a G to a C, now we have a G to a T. And then you can see the methyl group right here that was added, disrupting what was once a hydrogen bond there. And this is common in cigarette smoke, which is why cigarettes are a carcinogen, because they're effing with our DNA. Okay. So thymine dimers, like we said, from UV light, and it's a covalent linkage between two thymines right here. So we can have two versions of this. We can have the cis configuration and the trans configuration. And then also note these other names that they have right here. So cis configuration, C with C, that's the cyclobutane. And then I don't really know how to correlate trans with this, but maybe you can just remember that trans is the other one. But he uses these names later on, so that's why I'm saying to like kind of keep that in mind. So the trans configuration disrupts the helix much more than the cis configuration. So we're creating a greater distortion in our DNA double helix. So not good, but since we are creating more of a distortion, we're going to be able to repair that distortion more efficiently, because the proteins that are looking for these helix distortions can recognize this more easily than the cis configuration. So they have no problem with seeing that this DNA is effed, but they have a harder time like recognizing this cis configuration. So the disruption is more minor, but repair is not as efficient as the trans configuration. Okay, and then he's going into what some of the sources for DNA damage are. So that's going to be for radiation. We have UV, X-rays, beta emissions, and gamma rays. And then we also have chemicals, especially methylating or alkylating agents. So that's what we were talking about over here with the cigarette smoke. So it's a source of DNA damage. Don't smoke, guys. Don't vape. Not good. So first we're going to go into some repair mechanisms that are keeping the helix intact. So we're not breaking this DNA helix. We're keeping it intact. There's only two, and we only really care about one of them because this photolyase is not found in mammals. So cool, it exists. That's awesome. Maybe some researcher, like doing research on plants or something, needs this, finds it interesting, uses it for his research. Awesome. We don't really necessarily care because it doesn't have anything to do with humans. So we're going to focus on this MGMT. And I'm just going to keep referring to it as MGMT because this is a mouthful. So it's very important in humans, and it removes methyl groups. So these methyl groups, for example, if you're smoking cigarettes and they get added to your DNA, your MGMT is working overtime to take those methyl groups off. Okay, so this slide is basically showing you what cancer cells can do to basically like manipulate this MGMT. So cancer cells can develop easier if there is no MGMT present. So like this repair mechanism, these proteins involved in this repair mechanism would be silenced in some cancer cells. If they figure out how to silence those genes, then they can acquire more mutations and become an even bigger, badder, scarier cancer cell. So it might be favorable for them to silence those genes. So now if we look here, we can see the regulatory region for MGMT is over here, and we have an HDAC bound. So if we remember from lecture 2, HDACs reduce transcription. So they remove the acetyl groups from the histone tails. Histone tails regain their positive charge, and they closely interact with the negatively charged DNA, so we have condensed chromatin. Since we have condensed chromatin, we can't access that DNA for transcription, so those genes are not being transcribed. We're not making those proteins. They're basically like silenced. And he said an HDAC question is going to like 99% chance be on your exam. So make sure you know the functions of HDAC, make sure you know anything they're related to, like this MGMT silencing, and make sure you know hats and stuff like that. So definitely go back to that in lecture 2. Then he also said we have this HP1 keeping our chromatin closed, and we talked about that in lecture 2 as well, and I added a picture of that slide in case you had forgotten. But main takeaway from this slide is if cancer cells have their MGMT silenced, they can acquire more mutations and, you know, keep growing out of control. So it might be favorable for them to have HDACs bound to those regulatory regions. So just like cancer cells can manipulate this MGMT, so can we. So not having the MGMT silencing those genes is sometimes correlated with a poor prognosis, like for hepatocellular and biliary tract cancers, because now they're just acquiring more mutations. So that cancer becomes worse, and then it leads to a poor prognosis. Often those cancers aren't noticed early enough to treat efficiently. So it's just like bad luck. But we can also manipulate that they don't have those MGMT genes by inducing a mutagenic therapy. So we can try to mutate and destroy those DNA molecules in those cancer cells. And since they have no repair mechanism, then they're effed. So those cells are going to die. So basically, you thought it was a good thing to silence your MGMT genes, but now we've just mutated your DNA so bad that you can't carry out basic functions to survive as a cell, so now you have to die. So some cells, like glioma tumor cells, are more responsive to this mutagenic therapy. Okay, that was MGMT, a repair mechanism that keeps the helix intact. So now we're going to go to repair mechanisms that involve hydrolysis of phosphodiester bonds in DNA. So now we're no longer keeping the helix intact. So if you look over here, we're just removing this methyl group with MGMT. So we're keeping this helix like still intact. But then now when we get into all of these different repair mechanisms, in some way we are disrupting that helix. So we have nucleotide excision repair, mismatch repair, base excision repair, and recombination repair. So nucleotide excision repair has two categories, and that's going to be our global genome repair and transcription coupled repair. So global genome repair has proteins that are just always scanning for helix distortions, regardless of if we're doing transcription, regardless of if we're doing replication. They're just always in the nucleus looking for helix distortions. Transcription coupled repair, you can guess, is coupled with transcription. So those proteins are riding along with our RNA polymerase, and they're checking to make sure that there's no helix distortions before we transcribe that DNA, so that RNA polymerase is not transcribing some mutation and leading to a mutated protein or anything like that. And they're very efficient. So then we have mismatch repair, and that's going to be associated with DNA replication. So it's part of the replication fork. So those proteins are, along with all of our other replication enzymes, and they are basically kind of doing the same thing as these TCRs, but instead of transcription, we're doing it for DNA replication, making sure that we are double checking our work, making sure that there's no mistakes. Then we have base excision repair. So that depends on hydrolyzing the glycosidic bond. That's why he spent a lot of time making sure that you knew the structure of a nucleotide, making sure that you knew where the bonds are, what the bonds are to, on what carbon of the sugar. So keep that in mind. And then we have recombination repair, BRCA2 is involved in, and we'll get into that more later. Okay, so now we're talking about NER. So this nucleotide excision repair, NER. And its two divisions on this next slide. So like I said, we have this global genome and this transcription coupled repair, those two subsets of that NER. So with global genome repair, we have a helix distortion right here, and then we have this XPC. This XPC identifies a distortion in that helix. This XPC is what is defective in a disease called xeroderma pigmentosum, which is a sensitivity to sunlight. So XPC then recruits TF2H. TF2H is the last member of our pre-initiation complex that gets recruited, the last one. And it's a helicase basically for transcription. So it's opening up that DNA to allow it to be transcribed. So it's unwinding our DNA here so that we can get rid of like a thymine dimer, which is causing our helix distortion. And then from there, we have some phosphodiesterases that are cutting the strand. Then we have DNA polymerase delta, pCNA, and DNA ligase coming in and synthesizing our new strand and ligating everything together. So he wants you to know XPC. There are other XP proteins, but he wants you to memorize XPC. But then he said, so he said only memorize XPC, but if there's a question on the exam, he's probably not going to do that to you, but he's just saying if there's a question that says, which of the following proteins is important for xeroderma pigmentosum development, and there's only one XP something, and it's not necessarily XPC, then you know that that's going to be the protein. Anything with XP whatever is going to be correlating with this xeroderma pigmentosum. So that was global genome. So now let's look at transcription coupled repair. So transcription coupled repair, you can see we start off a little different up here and then we end with the same result down here. So we're starting off a little bit differently. We are recognizing our helix distortion with this CSB rather than an XPC. Then we also get this CSA is also recruited, and then those are recruiting our TF2H. And then from there, it's the same process with phosphodiesterases, DNA polymerase delta, pCNA, and DNA ligase. So main difference here is that for global genome, we have XPC recognizing the helix distortion. For transcription coupled repair, we're having this CSB, and that's both of those, that XPC, that CSB, whatever, are recruiting our TF2H. So same function as XPC, just different protein. So this CSB is riding along with our RNA polymerase. You can see RNA polymerase right here. So this is going to be happening when we are transcribing our DNA, whereas global genome repair is just happening all the time. Then he says CSB and CSA are unlikely to be correct answers on the exam, but XPC will almost certainly be a correct answer on the exam. So make sure you know XPC. Okay, and now these slides that he has here are his summary slides for like each of the repair mechanisms. And then he said the points of emphasis are in red. So he wants you to know global genome repair. He wants you to know global genome repair. He wants you to know it's a subset of NER. He wants you to know XPC. So XPC is recognizing that helix distortion. And these are why I asked you to pay attention to those other names for the thymine dimers, because this 6,4 photo product, that's the trans thymine dimer, and that's distorting the helix much more than the cyclobutane thymine dimer, which is the cis. So the 6,4 product is repaired much more efficiently. So the trans is repaired more efficiently since it's causing a greater distortion. It's easier for the proteins to recognize that. So then now we have our TF2H. Well, now we have our TF2H that got recruited. So we didn't really talk about all of these subunits and stuff, so I wouldn't worry about that as much. Just know that TF2H is being recruited and it's a helicase, so it's like unwinding that DNA. So we can see here in the image TF2H, then after TF2H did its job, now we have like an unwound DNA segment. So this XPG hydrolyzes a phosphodiester bond. I don't think he wants you to memorize this XPG. I think this hydrolyzes a phosphodiester bond is in red because now we are doing repair mechanisms that are involving the hydrolysis of a phosphodiester bond. So we're no longer keeping our helix intact. But if you do know it's an XP protein, then it has something to do with this global genome repair and this xeroderma pigmentosum. Yeah, because now he's getting into like XPF hydrolyzes the second phosphodiester bond. So you don't need to know those other ones. He said only XPC. Then we have PCNA. PCNA facilitates the processivity of DNA polymerase delta. So then we have DNA polymerase delta coming in and synthesizing our new strand. And then ligase is connecting that newly synthesized strand to the old strand. So that was for global genome repair. So now let's look at our little summary for transcription coupled repair. So it is also a subset of NER. So we know that it's with transcription. So RNA polymerase II is stopped by DNA damage. Then the proteins that are recognizing this damage to start the repair are CSA and CSB. They're basically substituting XPC. They're substituting for XPC in the global genome repair. So the XPC in global genome repair is recognizing the helix distortion. But for transcription coupled repair, it's CSA and CSB that are recognizing the distortion. So basically like the same thing.
Hey, Ivy.
Yeah.
So how it says like XPC and then HHR, do we have to know that part of the complex or just XPC?
Just XPC.
Okay, thank you.
You're welcome. So then in our global genome repair, XPC is recruiting our TF2H. So in our transcription coupled repair, our CSA and CSB are recruiting our TF2H. And then the remainder of our transcription coupled repair is identical to our global genome repair. So if I go back to the image, we see that here. Once we have recognition of our helix distortion, our XPC recruits TF2H, our CSB and CSA recruits TF2H. Once TF2H is recruited, then we have the same process. So as I was mentioning, he wants you to know XPC. So XPC is defective in xeroderma pigmentosum. We can see we have some other XP proteins. Those are also defective in xeroderma pigmentosum, but he just wants you to know XPC. But if we see xeroderma pigmentosum, we can assume any XP protein. And that's easy enough to remember. XP for xeroderma pigmentosum. X and P. So then we also have another syndrome that we're talking about, this Cockayne's syndrome. So CSA and CFB are the proteins that are defective in that syndrome. And we can think of that as CS for Cockayne's syndrome, C and S. So we have XP for xeroderma pigmentosum, CS for Cockayne's syndrome. And he touched on that the XP proteins are using an endonuclease that is cleaving out that DNA section from the middle, and we're not like in contact with the end of a DNA, which that would be an exonuclease. So we'll touch more on that, but if I go back to the image, we can see that this helix distortion is like in the middle of our DNA, so we have to use an endonuclease to cleave out that section rather than an exo. Okay, so now he's giving you like some symptoms of this disease, the xeroderma pigmentosum. Any disease that he gives you, you should know those. Like Bloom syndrome, we talked about all of the diseases in this lecture. I'm not sure if we've gone over more, I can't remember. I do remember Bloom syndrome from like lecture 3. Either way, when you're studying, when you're going through your slides, if you see a disease or like a disorder or something that he touched on, I would go back and make sure you know those. Because like I said, since this is a medical science master's, he would be inclined to put those questions on the exam to try to correlate it to medicine as much as you can. So make sure you're looking at those diseases. If he was going to ask you a question on this, he might say like simply XPC is defective, or he might say xeroderma pigmentosum involves a defective what? And then XPC could be the answer. Or he could say a patient is suffering from severe light sensitivity, early onset of skin cancer, severe pigmentation irregularities. What is defective? And then you would have to say XPC. So you definitely should know some of these like major symptoms, because he might give you the disease name or he might not. So make sure you know some of these like pertinent symptoms like severe light sensitivity, early onset of skin cancer. So then now we go to this other syndrome, and I highlighted some important things there. So like dwarfism is what's going to set this apart from this first one we were talking about. This xeroderma pigmentosum doesn't have dwarfism, but this Cockayne syndrome also has light sensitivity, has some neurological abnormalities, facial and limb abnormalities, and then early death due to neurodegeneration. So make sure you know the difference between those two. Make sure you know the proteins that are defective and what repair mechanism those proteins are associated with. Okay, now we just went over nucleotide excision repair. We went over both of those, so number one is done. He kind of skips mismatch repair, and now we're going into base excision repair. So we just did number one, and it's two subsets, so now we're going to do number three. So I wrote it up here for you guys, base excision repair. So base excision repair is most important when we have a uracil appearing in our DNA double helix. And we know uracil is not supposed to be in the DNA. Uracil is for RNA, so okay, that poses as a problem. So uracil is appearing in the DNA because we have a problem with deamination. We have deaminases in the nucleus, which makes cytosines more sensitive to losing their amino group. So if we're losing our amino group, that cytosine is basically becoming a uracil. This can also happen when we have that variant nucleotide that we talked about in lecture 1. If that loses its amino group, then it's becoming a thymine. But not much can be done about that, because when the proteins are scanning the DNA, there's nothing suspicious about a thymine because that's a normal nucleotide. So it's recognized as normal. But when we have a uracil, we're like, okay, that doesn't belong. This is not RNA, this is DNA. Let's cut that shit out. So we have uracil DNA glycosylase to do this. So that's the enzyme that's targeting the glycosidic bond. So now if we look on this slide, we can see the bonds. So we have uracil DNA glycosylase cutting out this this glycosidic bond. And we know the glycosidic bond is linking our base and our sugar. So if we look on the right side now, we can see that that bond is cut. That base is just free, I guess, just like gets dealt with later. So yeah, that uracil DNA glycosylase is cutting that glycosidic bond. We're getting rid of that uracil. Then now we're left with this weird phosphate and sugar and another phosphate with a new nucleotide. But our uracil's gone, so now we don't have a base connected to this sugar. So we have to figure out, okay, now what do we do? So we're going to cut our phosphodiester bond to get rid of that weird sugar without a base, and that's going to be catalyzed by AP endonuclease. So this AP endonuclease is cutting our phosphodiester bond, whereas our uracil DNA glycosylase is cutting our glycosidic bond. So we can see that also illustrated on this image. So, okay, here's our uracil. He circled it for you. So we have the loss of our uracil because we have uracil DNA glycosylase. So now if you look at this next image, we see that we don't have our base. So loss of the uracil, but we still have our phosphodiester bond. So we still have those bonds, those phosphodiester bonds, but we don't have a base. So our AP endonuclease is going to take care of that for us. So if you look here, we're cutting that phosphodiester bond. Then DNA polymerase beta is like removing that structure and that structure of sugar and no sugar, phosphate, no base. It's removing it. Then resynthesizing our normal Watson-Crick base pairs. So this is such a strange process that we have a new DNA polymerase. That's DNA polymerase beta, beta for BER. Kind of think about it like that. Don't get them confused with the other DNA polymerases that we learned about. We learned about alpha, delta, and kind of epsilon. So DNA polymerase beta is for BER, B to B. And then we have DNA ligase coming through and ligating it together. Then he said make a list of the order, but if I were you, I would maybe just make like a flow chart of what's happening in the order that's happening, and then after I make a flow chart, maybe off to the side, I would write what everything is doing. So if you make a flow chart, you can say like deamination. Well, you could have like cytosine, deamination to uracil. Then you could have uracil DNA glycosylase. Then you could have an arrow to missing base. Then you could have AP endonuclease. And then you could have an arrow to DNA polymerase beta, and then another arrow to DNA ligase, and then done. Then off to the side, you can say like what everything is doing, like uracil DNA glycosylase is cutting the glycosidic bond, taking away the uracil. AP endonuclease is cutting the phosphodiester bond. DNA polymerase beta is resynthesizing our normal Watson-Crick base pairs. DNA ligase is ligating everything together. And this AP endonuclease is also an endonuclease because we are not near the ends of our DNA. This is happening like in the middle of a DNA sequence. So here we have our summaries again. So our DNA glycosylase is remove base, and note how he says base, not nucleotide, because it's only removing the base, which was a uracil. It's not removing the phosphodiester bonds and the sugar, just the base. Then we have our AP endonuclease coming in and hydrolyzing the phosphodiester bond. Then we have DNA polymerase beta, which is removing the leftovers and resynthesizing the missing nucleotide with proper Watson-Crick base pair. Okay, so now we're getting into mismatch repair. So if we go back to our list, we just did base excision repair. We already did nucleotide excision repair and its two subsets. So now we're going to do mismatch repair. For nucleotide excision repair does TTR and TCR always happen on the right and left sides. I'm not sure what you mean by that, Crystal, because this is just a diagram showing us like what's going on, and they just like put it together where global genome repair is happening all the time. Those proteins are just looking for damage in the DNA all the time, whereas transcription coupled repair is only happening when we're doing transcription. Does that answer your question? Yeah, so this is just like trying to put it into like one cohesive diagram. So it's not necessarily always going to look like this. Okay. So now we're getting into mismatch repair. So mismatch repair is defective in this HNPCC. This HNPCC is a hereditary non-polyposis colorectal cancer. He has memorized under that. Oh, I didn't even realize that said memorize. Okay, you guys need to know this. I mean, I went over this in my session yesterday. Hopefully you guys when you're reading it, you see that, but that says memorize, so commit that to memory. But this hereditary non-polyposis colorectal cancer can be inherited for hereditary, or it can happen sporadically. You could just get non-polyposis colorectal cancer. So that was the basis of the case in the beginning of the lecture. They were talking more so about sporadic. So I guess this MMR is defective in either hereditary or sporadic colorectal cancer, which you learn more about HNPCC you learn about it in pharm, because you learn about its drugs in pharm, and then you learn about it in pathology. So that doesn't go away. So just keep that in mind. Okay, so we have an error. You can see that depicted by these little bulges in the DNA helix. And we can see this red line is not cohesive. We have a break right here. So this is representing an Okazaki fragment. If I go back to our list, then you can see that I said mismatch repair is associated with DNA replication, and it is part of the replication fork. So we have just replicated. Okazaki fragments are for the lagging strand. So we can still see an Okazaki fragment right there. It hasn't been ligated together yet. So the error is noticed before those Okazaki fragments are linked. And that's how the repair mechanisms know which strand to repair. So if we are synthesizing a new segment of DNA, then it's likely that, DNA polymerase could have made a mistake. DNA polymerase is not perfect, like all the rest of us. So it could have made a mistake. And we want to fix that before we're done replicating. So it knows not to repair the template, which is the correct sequence, because if it repairs the correct sequence and takes it out and puts in whatever the proper Watson-Crick base pairing is, then now we have an altered DNA sequence. So we just want to correct that newly synthesized strand. So that's how they're able to tell which strand is newly synthesized, because the Okazaki fragments are not yet linked. And since we're doing DNA replication and those Okazaki fragments are not yet linked, we can use an exonuclease this time because we have ends of our DNA that we can reach. So we can access those ends because they're not linked yet, so we're going to use an exonuclease rather than an endonuclease. So the protein that is recognizing the distortion in our DNA helix is going to be MUT-S. So it's recognizing that helix distortion. Then it recruits this MUT-L. So MUT-S and then recruits MUT-L, and then MUT-L recruits the exonuclease that's actually going to be chewing back the Okazaki fragments. So then now you can see we are going to chew it back, basically like backspacing what DNA polymerase did. So now we're left with this open segment. So then we're going to have, again, DNA polymerase delta, pCNA, and ligase come in and fix that DNA. So it's basically like a proofreading mechanism. We're going to backspace the DNA that was wrong. Hey guys, let's make sure our mics are on mute. Okay, so this MUT-S has subunits, and so does MUT-L, and this MSH6 is a subunit of MUT-S, and that was what was talked about in the case. And then this MLH1 is a subunit of MUT-L, and that's what contacts the endonuclease, or I'm sorry, exonuclease. So is MUT-L the exonuclease, or does it recruit an exonuclease? No, it's not the exonuclease itself, but it is like contacting it. I know like this blue thing looks weird, but it's not the exonuclease itself. It's like recruiting the exonuclease. Okay. You're welcome. So here is our summary in red. Okay, we're talking about mismatch repair. It's associated with base pairing errors that occur during replication. When we had talked about NER, that was associated with DNA damage. So this is more so like a proofreading mechanism. It's associated with colon cancer. That's important to know. And we recognize the distortion with MUT-S. Then we have binding with MUT-L. Then, we have excision by our exonuclease. And then we can also resynthesize that DNA. We get pCNA. I wrote it for you guys here. We get pCNA, DNA polymerase delta, and ligase coming in there and like fixing that strand. So then MUT-S has a subunit MSH6. That was what the subject of the case was. MSH2, not covered in class. Then we have MUT-L. And this MLH1 is frequently defective in HNPCC. If you remember, HNPCC is that hereditary non-polyposis colorectal cancer. So definitely know that. He has it in red here. He has memorized under it there, so make sure you know that. And then can you please explain the MUT-S and MUT-L slide again? Well, this one or on the diagram? On the diagram. Okay. So we have an error in our newly synthesized strand. DNA polymerase made a typo, basically. And these repair proteins are recognizing which strand is the newly synthesized strand because we have still Okazaki fragments that aren't linked together yet. Because if they were linked together, there's no way for those proteins to say which one is the newly synthesized strand or not. So we don't want to alter our template. We just need to fix the mistake that was made in our newly synthesized strand. So they look for those Okazaki fragments. Okay, those are still in fragments. That's our newly synthesized DNA strand. That's the one we need to repair. So then mu-S recognizes that and then recruits mu-L, and then mu-L recruits an exonuclease. That exonuclease chews back the Okazaki fragments, like backspacing to the typo that DNA polymerase made. So now we're left with this gap here. So then we have DNA polymerase delta and PCNA. PCNA gives DNA polymerase delta its processivity. And then we have synthesizing of our new DNA, the correct Watson-Crick base pairs, and then ligase comes through and ligates all of it together. Is the binding of PCNA at the mismatch just for preparing for the DNA synthesis before the exonuclease cuts the Okazaki fragment? PCNA is just PCNA is, so I guess you're maybe like asking about this point. If we go back to our lecture 3, whatever, we know that PCNA is what's giving DNA polymerase delta processivity, and that's basically all he wants us to know about PCNA. So I guess it's binding to the proteins that are recognizing like this mismatch right here, but I don't know. I'm just, knowing that PCNA gives our DNA polymerase delta its processivity. I don't know like why it's binding to like mu-L, but I do know that we need it to give DNA polymerase its processivity to like go through now and repair all of the DNA that we just like deleted basically. So does DNA polymerase beta have processivity too? No, I don't know if DNA polymerase beta has processivity. Yeah, we talk about DNA ligase, talked about DNA polymerase, but we don't talk about PCNA here, and we're literally only doing one nucleotide. So I would assume it doesn't have processivity. But that might be a good question to ask him.
Hey, Ivy, Dr. Blank said it doesn't.
DNA polymerase beta?
Yeah, he said in his lecture it doesn't, and I have it written in notes.
Okay, yeah, exactly. Thank you for that. So yeah, probably because it's only doing this one nucleotide, like I was saying, that's why it doesn't have processivity. DNA polymerase beta, for our purposes, is only important to know that it's working in this BER. Only DNA polymerase delta has processivity with binding to this PCNA. That's why we need polymerase delta, and we can't just keep going with DNA polymerase alpha. And then DNA polymerase epsilon is just like redundant. Okay. So I heard a student ask him in class, like, how do you get cancer from having defective MMR? We don't really know. If we did, maybe we'd be able to treat that cancer more efficiently. So maybe one of you guys can find out in your lifetime. But now we're moving on to recombination repair. So if we go back to our list, he loves his lists. So we did nucleotide excision repair. We did both of those. We did mismatch repair just now. We already did base excision repair, so now we're going to go to our last one, which is recombination repair. And we're keeping in mind that all of these involve hydrolysis of phosphodiester bonds, so we're not keeping our helix intact. Now we're getting to the last one. Okay, and then I wrote it for you here that this is recombination repair. So recombination repair is also important with DNA replication. So we just talked about MMR with DNA replication. But what makes recombination repair different is that we're dealing with a double strand break. So the issue now becomes, how do we fill this gap? Because all of the other repair mechanisms has been a single strand issue. So we always had a template to use. If we go back and look at those diagrams, I can show you what I mean. So here's our error made in our newly synthesized strand, but here we have our orange, which is our template, so we can synthesize this red DNA over here with the template of our orange DNA. So single strand, we have our orange as our template. If we go back here, okay, we excised our uracil, but we still have our template, which is a G, so we correct that and we put a cytosine there to properly Watson-Crick base pair with that guanine. So single-stranded issue, we still had a template. And then if we look at our XPC and our CSB, we're recognizing a distortion in the helix right there at the top, but although our TF2H is opening it up and we're cutting that out, we still have a template on the bottom to resynthesize that top strand there. So all of those have been a single-stranded issue and we still had a template to use, but now we have a double strand break. So what the heck do we do with this gap? Well, so what we're going to do is have chewing back unevenly between those DNA molecules. And then now that we've chewed them back, one of those DNA molecules can invade a neighboring DNA molecule. So we're in the nucleus, there's lots of DNA in our nucleus, so it's just going to reach over to its neighbor and be like, SOS, I need help. So it reaches over to its neighbor, we form a triple helix. So we have both of the red DNA strands and then now our invading orange DNA strand, so triple helix. So in that triple helix, it's basically using that neighboring DNA molecule, the red one, it's using it as a template. So we can see here that we're getting extension past where we had that double strand break. It's getting extension past there. And then once it gets extended past that break, then it goes back to its free DNA molecule. And it's like, okay, I have the template for us, like let's fix ourselves. Then we resynthesize from there. And we can properly Watson-Crick base pair from there. So just to reiterate, we get chewing back unevenly of the strands. One of those strands, it uses that molecule as a template, it gets extension, goes back to its buddy, and it's like, okay, I fixed it, let's resynthesize from there. So when we're forming this triple helix, we need BRCA2 and RAD51 to keep that triple helix stable. That triple helix is not energetically favorable, so BRCA2 and RAD51 are keeping it stable. So if we don't have BRCA2, that broken DNA will just be like floating around the nucleus and trying to recombine with other chromosomes. Then we can make new chromosomes that could be cancerous. We can destroy other chromosomes and the cell will die. So not good. So here I have it written, Rad51, BRCA2 help form the triple helix so that an invading strand of DNA can take advantage of a template from its neighbor strand. So now we have our summaries. So in red, BRCA2, he really wants you to know BRCA2. He said out of these two, Rad51 and BRCA2, remember BRCA2. So it's helping keep that triple helix stable so that the invading strand can take advantage of a template from its neighbor. Okay. So he's giving us another disease here, ataxia. I'm just going to call it ataxia. So we get DNA damage, and we can either get DNA repair in the cell lives happily ever after, or we can get failure to repair that DNA. So then if that DNA is not repaired, we get to a checkpoint that leads to apoptosis because that DNA is damaged beyond repair. That cell can no longer properly function. So then it has to apoptose. But if we have checkpoint failure and that cell does not apoptose, that can lead to cancer. And there is a disease, ataxia, where we inherit a defective checkpoint protein, and that makes people with this disease more predisposed to develop cancer. So it's hereditary. You can inherit this. Okay, there's your fill in the blanks. Do we have any questions before I get to your questions for the lecture? Are you going to post this PowerPoint on Canvas? I didn't see it up. I'm pretty sure I already did. Let me double check. Yeah, it's published. It's there. Okay, I guess there's no other questions, so then let's get into this. As always, put your answers in the chat. I'll give you a second to type them, but put your answers in the chat so I can see if we're understanding or what we're confused on. Okay. So which repair mechanism requires the use of BRCA2? Okay. Looks like you guys are getting it. It's going to be that recombination repair. So he might show you those repair mechanisms like this on the exam, just using like their abbreviations. So I would know what each letter like means. Like, so then you know like BER is base excision repair. So make sure that you know that. So which DNA repair method is coupled with transcription? All right, good. So that's going to be TCR, which TCR literally stands for transcription coupled repair. So that's what I was saying. Like he might give you a question like this, and if you know what TCR stands for, then you're like, duh, that's the obvious answer. But if you don't remember what TCR stands for, then you're kind of screwed for answering this question. So that's why I would say to know these like abbreviations, because it gives him more ways to ask you questions on the exam. So Cockayne syndrome is a disease related to which defective protein? Okay, good. That's going to be CSB. So, like I said, C and S, CSB. A patient is suffering from neurological abnormalities, dwarfism, and light sensitivity. Which disease do they have, and what is defective? Okay, good. That's going to be this CSB and Cockayne syndrome. You could tell that apart because I included dwarfism. So you can tell that apart from xeroderma pigmentosum because xeroderma pigmentosum doesn't include dwarfism as a symptom. So I think this is the last one. What is cutting the phosphodiester bond in BER? Yeah, good. That's going to be that AP endonuclease. The deaminase is what is causing the issue. Uracil DNA glycosylase is what is breaking that glycosidic bond. AP endonuclease is going to be, oh, why did that get deleted? AP endonuclease is going to be cutting the phosphodiester bond, and then DNA ligase is what is going to be ligating all of it together. Yeah, that is the last question. Do you guys have any questions on these questions before we move on to the last article highlights? Okay, looks like no. Nope, not that. Is it important to know what does uneven chewing back of the strands? No, he didn't really like go into that. Is it even labeled here? It's just a nuclease, but he didn't really touch on that. What he wants you to know is the idea of this strand invasion forming a triple helix, and that's why BRCA2 is necessary to keep that triple helix stable. When we don't have BRCA2, we can't form this triple helix, and those that double-stranded break will be just floating around, and that's how we get like chromosome fusion and different chromosomal abnormalities, because these free ends without telomeres on either side are going to be just floating around trying to recombine with everything. Is this an endo or exonuclease? That is a good question. If it's a double-stranded break, I would say an exonuclease. But does he specify? He does not specify, just says nuclease. I wouldn't worry about it, but I would say exonuclease. And if you want to email him, that might be a good question to ask him, or I could try to look it up for you later. But he didn't really touch on it, so I wouldn't say it's too important. When does BER take place? Base excision repair take place. So that's going to be just, I guess, whenever this DNA recognizes that we have a uracil. So if we have this deamination occurring and we like get a uracil, then this base excision repair is going to take place. So I don't think it's necessarily during replication or transcription or something. I think it's just going to be whenever we get this deamination. Okay. So now we will go over the case, or I'm sorry, the article highlights. So again, he doesn't expect you guys to read this whole article, know this whole article, and be tested on this entire article. He just wants you to read those highlights because it correlates with what we're learning in class. And it shows you like the purpose of what we're learning. But again, sometimes you need to read a little bit before or a little bit after to like get some context of what that highlight means. And then you can take notes that look similar to like mine. These are uploaded though. But sometimes, you know, like it helps to just take your own notes. Like I upload these as like examples, but you can organize it in a way that makes sense to you. And then that's how you know the material. So evolution of the cancer genome. So we get this somatic evolution and it can lead to cancer because we're talking about adaptations such as increased proliferative, angiogenic, and invasive phenotypes. So if we're increasing proliferation, we're getting our angiogenic properties, our invasive properties. That's what cancer is. Those are hallmarks of cancer. And those somatic evolution in our cells is due to alterations in the DNA sequence that have accumulated in the genome of the cell during that patient's life. So like if I was stupid and I went to the beach without sunscreen every day, I'm getting tons of UV damage to my DNA and I'm creating tons of thymine dimers, then that can account for somatic genomic alterations in my DNA just accumulating over my life. Maybe I just don't like to go, maybe I just like to go to the beach and don't wear sunscreen. And maybe I like to smoke cigarettes. So those are all accumulating in my DNA. So that is like the somatic evolution. Okay. So I said this in my review last night, but I'm saying it again. One of you guys asked me a question on the Friday Q&A about one of Dr. Blank's practice questions, and the answer was cancer. It was like something to do with massively parallel sequencing has allowed us to do what? And it was like the answer was cancer. And I told you that the answer was cancer, but I told you I couldn't remember where I had read that. So this is where I read that. The advent of next generation sequencing technologies has stimulated rapid cataloging of all alterations in cancer genomes. So if you're in my session, I hope I hope you're here now, or I hope you were there yesterday. That was what I was talking about. So we know that next-gen sequencing is massively parallel. So when I was talking with him, when he was reviewing the practice exam with me, he was saying he doesn't like the term next-gen, basically, because every time they came out with like a new sequencing method, it was like, okay, first gen, we had like our dideoxy Sanger, then second gen, we had our Illumina sequencing, and then now we have next gen sequencing. But starting with our second gen sequencing, which is our Illumina sequencing, they're all massively parallel because we're running more than one transcript at the time. So he said, when are the generations going to end? Because basically, we're just going to keep having new generations. They're going to keep finding probably better sequencing methods. So those generations are not going to end. Which, okay, yeah, I get it. So he just likes massively parallel. So if you see next gen, massively parallel. So that has allowed researchers to rapidly catalog all of the alterations in cancer genomes. So everything that is different in a cancer cell is now like cataloged. So researchers can look at large-scale genomic events or small-scale events. So the large-scale events would be like chromosomal lesions, copy number variation, and then the small-scale events would be point mutations and insertions and deletions. Okay, and then, so when I was reading this, it starts off with saying, for instance, the total number of cell divisions, whatever. So I was like, for instance, what? What is this an example of? So that's why I read the title of this little section here. And it says, the number and patterns of somatic alterations vary dramatically across cancer types. So I wrote that here, number and patterns of somatic alterations vary. So then now it makes sense, the example that they're giving us. So those alterations are going to vary. So like the total number of cell divisions in an adult lung cancer cell is greater than that of a childhood medulloblastoma. Makes sense. If I'm an adult, I've had more cell divisions, I can acquire a lot of mutations somatically throughout my life. I can have not mutations, but like mistakes in DNA replication and stuff. If it's a lung cancer, a lot of adults smoke, so I can have carcinogens. Versus like a childhood medulloblastoma, that's going to look a little bit different. So numbers and patterns of alterations vary. Then they give us more examples. So for instance, the colorectal pancreatic cancer cells with mutations in MUT-L have an increased frequency of nucleotide changes. That makes sense because we know, as we just learned in the lecture today, MUT-L is involved in DNA repair. So if we're not repairing our DNA, of course we're going to have increased frequency of nucleotide changes because we're not repairing those mistakes. So colorectal pancreatic cancer cells with a mutation in MUT-L have higher frequency of nucleotide changes. And then, okay, for this, so there's a relatively limited number of reoccurring alterations in and across cancer types. So basically, there's never going to be a magic bullet, one size fits all cure to cancer. It's just not going to happen. Cancer cells can be different within the same tumor. So I can have a tumor and it has like 3 different types of cancer cells in that tumor. So now physicians are having to figure out how are we going to treat all 3 different types of cells in that one tumor, in that one patient. And then my tumor might look different than somebody else's tumor. So basically what this is trying to relay to you is that the changes in the sequence of cancer cells is not the same. So we have this PIK3CA. That's the most frequently altered gene in breast cancer, but then only 20 6% of all breast cancer samples have that mutation. So to put that into perspective. Then the STK11 is the most frequently mutated gene in cervical cancer, and that's only present in 14% of samples. Okay. And then tumor cells are heterogeneous. They may contain a large number of individual clones, so they can have like different types of cells with different genetic alterations, different epigenetic alterations, different mutations that they've acquired. So tumor cells are not all the same. And breast cancer tumors are highly heterogeneous. So that's it for this article. Does anyone have any questions regarding the article? Does anyone have any questions in general?
For the article, should we focus on the examples or more of like the main picture that with like the adult and child? I was really trying to grasp it, but just understand that overall it makes sense that an adult would have more mutations.
Yeah, I don't think he would necessarily test you on I don't think he would necessarily ask you a question like which gene is the most frequently altered in breast cancer, and in how many samples was it present. I don't think he's going to ask you that. He's just probably going to ask you a question on the main idea to make sure that you're reading those articles.
Thank you.
You're welcome. He's a really fair tester. So he's not trying to trick you, but he wants you to read these articles. He's going to pull a question from the articles. Sorry if this was already stated, but approximately how many questions do you think will be from the articles? So in this exam block, you only have 2 articles, and they weren't that long, not that many highlights. So I feel like he's only going to pick maybe 1 question from these articles. I don't remember if it was exam block 2 or exam block 3 where we had more than this many articles, and I feel like he might have picked 2 questions from the articles. But there's not a lot that he can test you on with these articles, so it's just going to be 1 or 2 questions.
Hey, Ivy, I had a question unrelated to this, but back to the TF2D and the reinitiation complex. I was wondering how exactly the TF2D is related to the pre-initiation complex. Is it part of it, or does it help recruit it? I'm a little unclear about that.
So the TF2D is part of that pre-initiation complex, and it's going to be the last enzyme that is recruited to the pre-initiation complex, and it's a helicase that's going to open up that DNA so that we have access to the segment that we want to transcribe. So I don't know if it's exam block 2 or 3, but we learn more in depth of what the pre-initiation complex is, and I'm sure we'll go into it more, but it's the last enzyme recruited in that pre-initiation complex.
Oh, sorry, I said the TF2D, not the TF2H.
Oh, I'm sorry. Okay, TF2D, yes. Okay, let me pull that up and go through it with you guys. That was lecture 5 and 6, or lecture 3 and 4. I think it was 5 and 6. So TF2D, here it is. Okay, let me share my screen with you. So TF2D is part of this pre-initiation complex. It's going to be the first one that starts the pre-initiation complex. It's like a protein enzyme that's part of this. So it has this subunit for the TATA binding protein. So when we have methylation of our histones, the TF2D sees that and it's like, okay, this is where we're going to start transcription. Then its TATA binding protein binds to the TATA box. And then TF2D is recruiting all these other different transcription factors. And then it's also recruiting this RNA polymerase 2, which is the one that we use for protein coding genes. And then once RNA polymerase 2 is there, then we will get transcription. So it's part of initiating transcription. So it's like recruiting all those other transcription factors that we need at that gene to initiate that transcription. Does that make sense?
Yeah, that does. Thank you. I think I was just getting like too caught in the weeds of like
Yeah.
the exact, yeah.
Yeah, we get more into that later on in the course. So he just wants you to know those two things that he talked about, the TBP and recognizing that histone methylation.
Great, thank you.
You're welcome. So for the HDAC that we discussed while talking about MGMT, does that HDAC reduce the silencing? No. The HDAC is what is causing the silencing. So whenever we have an HDAC, we condense that chromatin. Since that chromatin is condensed, we can't access that gene for transcription. We can't access that gene for replication. So basically that gene is silenced. Does that make sense, Alexis? Yeah. So cancer cells, like, they want to acquire mutations, so they don't want those mutations to be repaired. So they're going to recruit those HDACs to the MGMT regulatory regions so that we can silence the MGMT gene, basically. Then we're not going to make any more of the proteins that are repairing the DNA. What methylates H3K4? Okay, so I uploaded this document about chromatin regulating proteins, and there's a histone methylase, HMT. So I'm pretty sure it's HMT that does that. Does anyone else have any questions on anything, like exam, previous lecture? I don't know, anything. Finished kind of early.
Ivy, yeah, I had another question. I was going to wait in case other people had questions, but the SNPs, I know that they're not associated with medical issues, like directly associated, but I think he mentioned something about like they can be used to track medical issues. I was wondering, like, I guess what we need to know about that.
Yeah, so the SNPs are not always implicated in like a disease, whereas like the OMIM alleles are disease related all the time. So a SNP is just like a variant nucleus or a variant sequence, I guess you could say. So like at one position, I might have an A, you might have a G, but that doesn't mean like as a fact that I have a disease. But if I have an A there and I have type 1 diabetes, and then every other person that has type 1 diabetes also has an A there, then okay, maybe that SNP does have something to do with a disease. So we can try to track those alterations, those SNPs, and see if they're similar in people with diseases. So it doesn't necessarily always have to relate to a disease, but it can be related to a disease. And if like everybody that has that disease has that variant, then okay, maybe this SNP has something to do with developing this disease.
Great, thank you.
You're welcome. Would you say for the genome browser, we just need to know where things are located, like based off your genome browser slides? Yes, I would say that. But you also need to understand like some of the concepts behind things, like how I just explained this SNP stuff. OMIM are variant sequences that are related to diseases. CPG islands are regulatory regions that are methylated. Like you need to know those concepts, and then you need to be able to apply them to like where they are located on the genome browser. Aside from genome browser screenshots, will he be likely to use diagrams or pictures in questions? Yes. So that's why I like harped on it. Know the difference between sister chromatids and homologs. Pull up a karyotype and count how many homologs are there, and then count how many sister chromatids are there, and make sure you can do that for every single karyotype image. Because I'm not sure. Yeah, I think we had like 3 pictures of karyotypes on my exam last year.
Hey, Ivy.
Mm-hmm.
Could we go over the questions that he sent us? I just didn't know what the answer would be for the first question. What's important to drive the nucleic acid synthesis forward?
So I didn't want to go over those questions in case people hadn't done them yet, because then it kind of ruins it for them. Let me. My iPad is dead. I don't have the questions pulled up.
Email you?
Yeah. So what I was going to say is I had a couple students email me their answers, and then I checked their work, and if they got an answer wrong, then I would provide an explanation of why their answer was not correct and why the correct answer is correct. So if you want to do that, then I will do that with you, or we can go over them in my office hours tonight.
Okay, I'll just email you.
Yeah, perfect.
Thank you.
Of course.
Hi, Ivy. I had a personal question. I didn't want to waste anyone's time, but I was just like, I don't know if like once the meeting ends, if I could ask you.
Yeah, sure.
Okay, thanks.
I'll stick around for a little while, and then if nobody else has any more questions, then yeah.
Hey Ivy, I have one more. I was wondering if the ideogram is specific to the BAC, or if that was also used for massively parallel sequencing to refer to the like overlapping.
The ideogram. Oh, like when we form contigs, is that what you're?
Or yeah, I meant contig, not ideogram.
Okay, yeah, the contigs. I'm fairly certain that the contigs are just with the bacterial clones. Because with the bacterial clone, that's when we get those large overlaps, larger than reads, and that's how we can be fairly certain. So that's how we can like piece together basically like an entire like chromosome, or an entire region of a chromosome, which is what we denote as contigs. Yeah, just associate contigs with the the bacterial clones.
Great, thank you.
You're welcome. You're welcome, guys. If there's no other questions, yeah, good luck. You guys are gonna do great. I have office hours tonight from 6 to 8, and then I'm gonna have like office hours tomorrow from 9 to 11 for like any last minute questions. Hopefully you guys are doing the practice exams. Please go back and look at all of those practice questions. Remember, there's like 220 practice questions for a 50 question exam. Like, do all of those practice questions.
Hi, Ivy.
Hi.
I have a question regarding the practice exam. For C1, you said that it's always going to be the one that has the sugar connected to the nitrogen space, the sugar.
The C1 position on the sugar is always going to have the base attached through the glycosidic bond.
Also, can I also say that the glycosidic bond is connected at C1?
Yeah, that's what's connecting the base and the sugar.
Okay, and then the phosphodiester bond where the phosphate is attached, that's C5, correct?
Yes.
Thank you so much.
You're welcome. Exon intron processing does not involve RNA molecules, correct. Exons and introns are RNA molecules. That's after we go through transcription. So I would say yes, they do. Any other questions, guys? You're welcome, guys. Good luck tomorrow.
Just a quick question again to clarify. The lysine 4 on histone 3, it must be present for TF2D to stop pre-initiation complex. Is that correct?
It needs to be methylated. So if it has that methyl group attached to it, then TF2D will recognize that and initiate the pre-initiation complex.
And it's specifically to the lysine 4 on histone 3, correct?
Correct.
Thank you.
You're welcome. I had a question, but I can't remember, so I will email you. Okay, sounds good. Will you be going over the additional and example questions posted? You mean the questions by Dr. Blank, Kara?
Yes.
I can go over that in my office hours tonight or tomorrow, or you can email me. I just didn't want to go over them right now in case somebody hadn't done them, because then that like ruins it for them. But yeah, if you want to come to my office hours tonight or tomorrow, we could do that, or you could just email me your answers and then I can provide you with an explanation of why something's right or wrong.
Thank you.
You're welcome. Oh, if you remember your question, Lucy. No? Okay. Does anyone else have a question? I cut somebody off.
Yeah, that's okay. I'm looking at the synthesis of primary transcripts and the splicing of exons. Excuse me. I'm a little bit confused about when we have, I'm looking at that picture, right? Because I think I got a question wrong on one of your tests. Oh, okay. It says that the translation, we have all the exons, introns out, and then we cleave out the intron. We have the exons down where the mature RNA are, right? But there are like two ends that has like 5' UTR and then 3' UTR. But when we have the translation in the new peptide, there is something that, there was a question about, there is something that's not included on that peptide. I'm not sure which one that's, yeah, that's where I'm confused. I'm not sure on the polypeptide one, we only have the exons, right?
Do you know which question you're asking about?
Yeah, this is the human genome one.
From my Friday Q&A questions?
No, this is from the lecture September 9, 14, 16, and 20.
From, it's like a question I asked or just the topic?
It's from the topic.
Okay, so the question is, what is included in the final mRNA?
Yeah, it says polypeptide, but because on the mature RNA, I see the 5' UTR and also 3' UTR also included on that strand. But when we go down to the translation, we have the polypeptide. I'm wondering, is it only the one to like the 3 blocks 1, 2, 3, which are the exons are included?
Yeah, so the
Yes,
5'.
Yes.
Oh, am I still sharing my screen?
Yes, it's showing and I see it. That's correct. That is exactly right. Yeah.
Okay, so 5' UTR and 3' UTR mean untranslated region. So they get transcribed when we're going from DNA to RNA. So they're going through transcription, but they're not going through translation. So we have them, so they're part of our DNA sequence, then we go through transcription. So we have our 5' UTR, our 3' UTR, then we're cleaving out our introns and splicing together our exons. So we still have our 5' UTR and our 3' UTR, but they're not getting translated into a polypeptide because they are the untranslated regions. So they're part of that mature mRNA, but they're not part of the polypeptide. They don't get translated. Can I go over the massive sequencing process?
Thank you so much. I got it now. Thank you.
You're welcome. Massive sequencing process again with the plate with plopping back and forth. Okay, let me pull that up. Am I still screen sharing? Can you guys still see my screen?
Yes.
Okay. So this is what we wanted to go over. So, okay, we have our DNA and we have primers. So we have fragmented our DNA into overlapping pieces, and we have attached primers onto those DNA sequences. So here we're adding the primers. We also have more primers covalently attached to this little chip, this little piece of plastic. We have all these primers attached, and our DNA is attached as well. So we have primer, DNA, primer. So those primers are like complementary. So they can like plop down and just like bend over, and they can Watson and Crick base pair. So then we get this little bridge. So then we start synthesizing a new DNA segment off that primer using that little bridge as a template. So from this primer right here, where the arrow is going, we're using this little bridge as a template, and we're synthesizing a new strand of DNA. So now we have a double strand of DNA here. So then we're going to denature that double strand, and now we have like DNA attached to the chip, and they're like next to each other. So here you can see 2, 4, 6, 8 when we only had 4 to begin with. So then we keep doing that, we keep doing that. Once we get complete amplification, we can fluoresce them and then take pictures, and then we get a picture like this. And then every time a new nucleotide is added, we take another picture, and then we compare, and then the computer like maps all of this out and then tells you what the sequence is. So I hope that answered your question. You're welcome.
Hi, Abby. Just to make sure I understand, for my mtDNA variants, I have the stop codons, the methionine, right? It's only methionine that says start codon, right?
Yeah, but that's not going to be relevant for this exam. He's not going to make you memorize those codons.
Oh, great. And also because there's like a V600E genes there too. Is it?
That's important. You need to know the V600E mutation, but you don't need to memorize all of the codons.
Oh, great. And that's the mutation for melanoma?
It's in the BRAF protein. Yes, it's in melanoma. Yeah, so we have, I can pull it up. So which lecture was that? This one? Here. So yeah, that's present in melanoma. It's in the BRAF protein. The BRAF protein is this pro-proliferative kinase. So if that gets mutated, we can get cancer, i.e. melanoma. We're switching from a valine to a glutamate. So glutamate is negatively charged like phosphate. And when the BRAF protein gets activated to proliferate, it gets phosphorylated. So like we said, phosphate is negative, glutamate is negative. It thinks that it's being turned on with a phosphate when it gets this mutation to a glutamate, since they're both negative. So it's like, oh, I'm phosphorylated, I must turn on now. So then it turns on, and then it keeps proliferating, but it doesn't stop because that negative charge is always going to be present, because that glutamate is always present there now. Whereas if we're phosphorylating, we can dephosphorylate and take it off.
Okay, thank you.
You're welcome.